Hey, Mr. Grove here. Today we're going to talk about some non-Mendelian genetics, which are kind of the exceptions to the rule. Gregor Mendel did a great job laying the groundwork for us, and when he studied his P traits, remember he chose those 7 specific traits to formulate his chromosome inheritance theories. And it really worked. He came up with those traits, and he said they were passed along and inherited independent of each other, which was true for him because those traits, he just so happened to pick 7 that were on 7 different chromosomes. And so they were inherited independently of that. And so that was really fortunate that he could learn the rules without any exceptions to those rules. However, we now know that there are lots of genes that are on the same chromosome as other genes. And so those oftentimes will be what we call linked together. And so these linked genes, like the example of red hair and freckles, are oftentimes passed along together. And it wasn't until this guy, Thomas Hunt Morgan, who came along in the 1900s, early 1900s, at Columbia University, and he studied the fruit fly. And so he did his genetic research with the fruit fly, and unlike Mendel, he did not have a wide variety of different fruit flies to start with. And so he just bred fruit flies after fruit flies after fruit flies, which they breed very, very quickly. And then I think it was like in 1910, he got his first mutation. The normal eyes of the fruit fly were red, and he got a white-eyed fruit fly. And so that was a huge breakthrough because now he has a different phenotype that he can start crossing. But when he crossed this white eye with the normal red eye, he would notice that white-eyed traits showed up more often in males and not in females. And so after further study, we realized this is what we would call a sex-linked trait. So a sex-linked gene is located on the sex chromosome, so either on the X or on the Y, most likely on the X because there's not as many genes on the Y. So, and that was the case. So to have a female fruit fly, which is possible with white eyes, he would have to have both the X copies would have to have that colorblind trait, whereas males, only one of the only X chromosome. If that colorblind trait is on there, then that male will be colorblind, or in this case, white eyes. And so the likelihood of sex-linked traits show up way more common in males because of that. So of the thousands of students I've taught in my career, we've had several colorblind students that are male. In fact, almost at any one given time, I will always have at least one student. One of my good friends who's a colleague and a really good ag teacher, Mr. Zimmerman, he is colorblind and obviously is also male. But in all of my teaching students, I've only had one colorblind female because statistically, to be colorblind, you would have to have that colorblind trait on both of your X copies. Just on one means you're a carrier, but you still have a good gene that would work. And so that is kind of how it shows up. Another trait, unlike colorblindness, is hemophilia. And so hemophilia is when your blood does not clot properly, and so this could be a very serious condition, and showed up in a lot of royal noble families in the 1800s and early 1900s in Europe. And so we saw Queen Victoria was a carrier, and then it ended up being passed along to the Russian family where Alexis was hemophiliac. This is also a good case where when you limit your gene pool, these traits can get stuck in there and are hard to get diluted out. And so in the 1800s and early 1900s, if you are a noble or rich person in Europe, you're probably not going to marry some random peasant. And so the amount of potential genes are definitely less available. And so that we saw that kind of happen with hemophilia getting caught in these kind of noble European people. So Back to colorblind. So red-green is the most common. So if you cannot see the 7 in here, then that means you are colorblind, and that would mean you would have on your XY, your X is going to have that colorblind trait, and that came from your mother. So Dad didn't have anything to do with your colorblindness. Let's take a look at a little Punnett Square. Okay, so Bob is not colorblind, so that means he's going to be XY, normal. Sue is not colorblind, but she is a carrier for the trait. So we want to know what are the odds they'd have a colorblind daughter, a colorblind son, normal-sided daughter, and normal-sided son. So if we do our Punnett Square, we would see it to look like this. And so colorblind daughter, statistical odds, we would have not here and not here. This one's a carrier, but still has a good copy, so 0% chance. Colorblind son, we have 1 of the 4 here that would be colorblind, so 1/4. Normal-sided daughter, both of these would be normal-sided, so 2 out of 4. And then a normal-sided son. So on this one, we would have 1 out of 4 again. And so that's how you would look at a sex-linked gene in a Punnett Square. Another exception to Mendel's rules is what we would call incomplete dominance. Okay, so if we were to cross, typically a red carnation with a white carnation, so big R, and big R, little r, we would expect all of them to be big R, little r. That's what Mendel would say. And we would expect them to have the dominant color of red. But when we actually do this cross, we get all of them to be pink. And then if we cross 2 of the pinks, we get a red, a white, and then 50% pink. So it's like this heterozygous trait is a blend between the 2 homozygous dominant and recessive colors, phenotypes. It's like the dominant is incomplete in its dominance over the recessive trait. And so that is incomplete dominance. When we have a blended color in between the 2 homozygous, we call that incomplete dominance. So let's take a look at an example here. So we have a brown guinea pig and a white guinea pig, and they get crossed, so something like this. And when 2 of the tan offspring are crossed, and so then they would have 8 tan offspring. So let's take a look at what our Punnett Square would look like. And we would see that, and we would see that, and we would see that. So, so we would expect these to be all brown under normal Mendelian circumstances, but the reality is they were all tan. And so that shows us that this is going to be an incomplete dominance. Okay. If we cross 2 of the tans together, so take 2 of these offspring, cross them, so we would get big B, big B, big B, little b, big B, little b, and little b, little b. And Mendel would predict this would be normal 3/4, 1/4. But the reality is we get, our probabilities would be 1/4 brown, 1/4 white, and then 50%, 2/4 tan. And so if we see, that's kind of about what we had here. Remember, these are just probabilities, so real life is going to probably be similar, but not necessarily exact. So that is how we can tell that what we had was incomplete dominance, because of that existence of the heterozygous being a blended phenotype between the 2 homozygous possibilities. Another kind of Mendelian exception is codominance. Okay, and so this is when both traits get shown. So if we have like white and a black chickens and they get crossed, then we would get speckled. Or maybe if we have flour where it's half and half. Okay, so it's not a blended phenotype, it's actually both of them showing up. So it's co-dominant. They're both showing their traits, their genes. So let's take a look at this. So we have a white tulip and a red tulip crossed. Okay, so their offspring would be spotted red-white tulips. So we have something kind of like this for their offspring. And then if we take two of those red-white offspring and cross them, our Punnett Square would predict something like this. And so what phenotypes would we expect to see in the next generation? Well, we should see some red. We might see some red-white spotted, and then we'd see some white. Okay, so the fact that we're seeing both traits and not a blend would tell us that we would expect this to be co-dominance, not incomplete dominance. And the fact that our heterozygous is different from our homozygous dominant tells us that it is one of the exceptions to Mendel's basic rules. So that's how you could look at a co-dominant genotype. Some other kind of exceptions would be the fact that sometimes we have more than two alleles. You know, we've been talking about eye color, brown and blue. We've been talking about, you know, albino or not, cystic fibrosis or not. But sometimes you have more than two alleles. And so blood type is a good example of that. So we have A blood type, B blood type, AB blood type, and then none of these carbohydrates, which would make it an O blood type. And so multiple alleles then can give us even more variety then of our potential phenotypes. Another thing that's not normal Mendel is this idea of Pliotropy. So sometimes lots of genes will influence, or one gene will influence lots of traits. Okay, so and that's the case with sickle cell. So we have sickle cell is just one gene, but there are all these different phenotypes that can be affected by that one sickle cell gene. So that would be Pliotropy. And the opposite of Pliotropy is kind of polygenic inheritance. And so we kind of talk about in class, you know, tall or short, as if it's one gene. But the reality is height is actually influenced by multiple genes, skin color is by multiple genes, hair color is multiple genes, and that's why there's so many shades. Eye color is also that way. So we have brown and blue, but then we get things like green and gray, hazel and almond, because of the shading that goes into that as well. So it's oftentimes a single phenotype, like color of skin or height, can be affected by lots of different genes, and so we call that polygenic inheritance. And so those are just some of the exceptions to Mendel's rules. Thankfully, Mr. Mendel in the monastery did not have to encounter those. He chose very well in his 7 traits that he could set the basis foundation for us in understanding genetics. And then 50 years later, Thomas Hunt Morgan came along with his fruit fly friends and were able to understand the exceptions to these rules a little bit for us. And this understanding of genetics is very valuable in our breeding of agricultural plants, livestock, and just understanding in general how traits are passed along from one generation to the other. So I hope that was helpful.